Yes, these are the neural components that we're going to be talking about now. The cells that are embedded in the retina. This is really what your retina is made of, these various kinds of cells. Some of them are receptor cells and some of them are nerve cells. The very posterior layer of the retina is an epithelium. It's neither of those. It's a pigment epithelium, and it absorbs light. It absorbs light and keeps reflections from confusing the receptors. For the scalps that we saw earlier in cats have behind their eye, they have a mirror-like membrane. So when light goes into their eyes, it reflects back out again, and the receptors get the light twice. So that's why cats can see so well in the dark. And that's also why when you shine a light in a cat's eyes, it reflects back out at you. So the photoreceptors are the next layer. They're derived from the same stem cells that produce ependymal cells. So these rod-like ones here, those are rods, rod cells. And they're basically your night vision cells. And the ones that look like cones here, the whole thing is a cell though. The whole thing is a cell. Just the top part looks like a cone. These are your cone cells. They detect color vision and light. So here's an image taken of the actual tissue. There's the sclera. I don't know if we talked about the function of the sclera. The sclera is a thickish membranous layer. It's the very outer layer of the eye. And there's a lot of fat around the eye as well, which helps it not to be jolted against the eye socket. But the sclera maintains the shape of the eye as well. And protects against friction against the eye socket, which is bone. And the next layer is choroid. And that is, we did talk about this, didn't we? It's vascular, it's got a lot of vascularization. So it provides blood to the retina. So now we're in the retina, this part here. And it is layers of pigment epithelial layer. The rods and cones are the outer segments. Here are the nuclei. That's why this area here is so dark. That's a concentration of the nuclei. And the next layer of cells are bipolar cells. They're nerve cells. And the next layer after that are ganglion cells, and they're nerve fibers that go straight to the optic nerve and become part of the optic nerve. And then below that is the vitreous body that helps to hold the retina against the choroid. So let's take a closer look. Why are they so good at detecting light? Part of the reason is that this rod cell here, the outer segment, is stacks of discs. Those are all discs of membrane. Like there could be 1,000 of them. And all of those are embedded with receptor proteins or opsins, which are pigments. And the cone cell has the same thing. These are also stacks. of membrane discs. The inner segment is the part that holds mitochondria and other requirements of any cell. It's quite a lot of mitochondria. And the nucleus for both is here. That's the cell body. And there's an outer fiber that synapses with the bipolar cells. So you can see in this micrograph that the rods are a lot larger and the cones are smaller. So can you think of a reason why the rods might be larger than the cones? Let me just put the chat up here. Why might the rods be larger? So remember, they're detecting, to be more sensitive because they're detecting light in essentially dark conditions. Yeah, exactly. So what the discs do overall is they increase the surface area of the receptor for pigments. And where are these pigments, you ask? They're embedded in the membrane. So here is just one of the discs. And the yellow bits all around are pigments. In this case, the pigment is a combination of a protein called opsin and a molecule called retinal, which is a derivative of vitamin A. And so this is what I mean by pigments. What happens to them when they get hit by light? They have two shapes that they can be, and they can conform to either shape. So this is the retinal molecule. But this conformation is called cis-retinal, where it's bent here. And this conformation is called trans-retinal, where it's straight. So this is bent and this is straight. And that's what drives the whole thing. That's what drives the conversion of light to electrochemical signals. Yeah, so I'm just scrolling up for a moment there on request. So those are the details of our rods and cones. There are cells that are non-receptor cells that we talked about. They are bipolar cells that are first order neurons. They synapse on the ganglion cells, which are second order. And there's a whole lot of convergence. Usually. Yeah, no, there is. Yeah, there's a lot of convergence between the bipolar and the ganglion cells. Yeah. So the axons of the ganglion cells form an optic nerve, and there's more convergence. There's other smaller, but very instrumental for vision cells called horizontal cells and amocrine cells, and they form connections right there in the retina. So right in the retina, your what you're seeing is already being processed with respect to contrast, edges of objects, and changes in light intensity. So I'll show you how it changes when we talk about the cycle when light hits the cell. So here are the rods and cones, and here's a horizontal cell here. That's closer to the junction between the receptor and the bipolar cells, and the amacrine cells are here, where the synapse between the bipolar and the ganglion cells occurs. So that really helps to start to process the perception of vision even before the signal goes to the brain. So the pigments of the rod cells is called rhodopsin. There's two major parts of that molecule, the protein called opsin and the vitamin A derivative called retinal. So the absorption peak, or the type of wavelength that rod cells can absorb, are 500 nanometers. So that has the most impact, but they also absorb wavelengths at either side of that. So here's the photochemical reaction of rod cells. So it starts off bent in the cis configuration. And here comes light. Boom. It changes that cis retinal. It isomerizes. That means it changes shape to trans retinal. And then because it's changed shape like that, it separates from the opsin. But in the meantime, it's triggered a reaction. I'll show you what that is in a moment. But it takes a little while for that retinal to get back to the opsin. So it takes 5 minutes to regenerate 50% of the rhodopsin, combining them again. Yeah. So this looks complicated. It's not too bad though. Let's look first on the left-hand side. This is in the dark. So in the dark, here you have your receptor. The receptor is here. And the most important thing is that this rod cell is releasing glutamate here. Releasing glutamate. So that's an inhibitory postsynaptic potential. It inhibits the bipolar cell from releasing any neurotransmitters. So that's the signal that it is dark. Yeah, so no signal is it's dark. But what happens in the light? When there's light. So This was a channel for sodium, and it closes. It closes, so nothing's happening there within that rod cell. Because it's closed, the cell becomes hyperpolarized. Because it's hyperpolarized, glutamate ceases. So no glutamate, that's the receptor, is released. So there is no inhibition anymore. There's no postsynaptic inhibitory signal. So the bipolar cell is not inhibited, and it releases the neurotransmitter here. When it releases the neurotransmitter, it starts a series of action potentials down here. The signal for light, the signal for light. Yeah, so that's the story of rods. And if you wake up in the middle of the night and it's bright, the pupils constrict immediately. That happens fairly quickly, but there can be some pain because it doesn't happen quickly enough for the light not to reach the retina. And so once the light turns on, your color vision and acuity is not optimal for a few minutes, for 5 or 10 minutes or so. So why do we have both rods and cones? So far I've only talked about rods. So rods are such that they detect small bits of light. And they can distinguish between shades of gray. Well, that's great, but they're not capable of distinguishing detail. Cones are. So a single type of receptor cell couldn't have both the high sensitivity of a rod cell and the high resolution of cones, the cone cells. Yeah. Night vision is very sensitive. It's one type of cell. Day vision is a different kind of cell. It has a high resolution and it has a pigment that detects color. Color pigments and a different kind of circuit. And you'll see what I mean in a moment, why daytime vision is so acute. Night vision is also known as scotopic night vision. Rods, very sensitive. So we've already seen that they're quite a bit larger than cone cells. So say this is 1 square millimeter of the retina. Here are all these rod cells, many rod cells, and they converge. So here we're showing 5 rod cells converging onto one, and all of these 3 converge onto this one. So convergence increases sensitivity to light, to dim light. And the rod cells they're spaced apart quite a bit. They're widely spaced. And it's a very low resolution, so you're not getting a lot of detail, but they do alert you to motion. Particularly in peripheral vision. The daytime system is called your photopic system, day vision. So there's only, in the fovea, that's where all your cones are, there's only about 4,000 little cone cells, no rod cells. And what's important, there is no convergence. So each cone cell here has a direct line to a bipolar cell here, which has a direct line to a ganglion cell. So there's very, very high resolution, like each one has a private line to the brain. So very high resolution, but not a lot of spatial summation, so not a lot of detection of motion, and less sensitivity to light intensity. Topic vision. So primates, that's us, we have very well-developed color vision. Nocturnal vertebrates, like raccoons, for example, around here, raccoons. In Australia, they're all nocturnal. The numbats are nocturnal. And they have a lot of rods. There's also in the Galapagos Islands, there's Galapagos seabird. I can't remember the name of it right now, but it's the only known nighttime bird, a nocturnal bird in the Galapagos, and it has loads of rods. So nocturnal vertebrates are those that function at night. That's when they go off and feed. So they need to be sensitive to light. They have only rods. But diurnal animals like us and other animals, diurnal animals have more cones. And interestingly, we have blue, red, and green cones, not the primary colors. And they're named that for the absorption peaks of their pigments, which are called photopsins, as opposed to rhodopsins, photopsins. And the blue cone's peak sensitivity is 420 here, the green 531 here, and the red 558 here. You can test yourself for red-green color blindness. So if you're looking at that right now, you should be able to see a number. What is the number? What is the number that you see there? Yeah, 16. Yeah, it can look a little bit like a 2, but if you're seeing green within the red, then yeah, you're good. Color blindness is genetic. So where the gene that codes for the red-green proteins that are part of the pigments is not functioning. So it's a recessive X-linked gene. So females don't tend to have it because we have 2 X chromosomes. So if we have the colorblind recessive allele, it doesn't matter if we have the dominant one. So the dominant is normal vision, and the recessive is red-green colorblind. There are different kinds of color blindness. But for males, if they get an X chromosome that has the recessive allele, the Y chromosome doesn't have anything to mask that allele. So males, I think I can't remember the number of males that have red-green color blindness, but it's actually quite a lot. But I don't trust myself to remember it. I'm thinking 1 in 8, but I don't think that's right. but it is definitely more common in males. So I just wanted to say a few things about color vision. The pigments are called a photopsin. Some really good notes somewhere. Where did they go? The cells themselves, the cone cells, have different amino acids No, hang on. Sorry. Cone cell opsins, proteins, have different amino acids that determine which wavelengths are absorbed. So that produces color vision. I've got it now. 8% of males, apparently. So approximately 8% of males exhibit color blindness. Yeah, so, but one thing is that a person that has color blindness makes up for it by being able to see contrast more, by being able to see the difference between shapes more. So like a friend of mine is a botanist and worked in the Sichuan area of China where there are biodiversity hotspots. And so he was looking at plants and recording the different kinds of species of plants there. And somebody he worked with was colorblind, but a botanist, which is interesting because some ways that you distinguish one plant from another is by color. And so they'd be driving along and his friend would say, oh yeah, that's a such and such plant in the distance, from a long way away. And it turns out that he really was very acutely sensitive to the shapes of leaves, for example, and other distinguishing features of plants other than color. So that was really cool. So one thing that we have, because we have binocular vision and our focuses can cross over, is depth perception. That's judging how far away objects are. So you have to have overlapping fields though. Horses don't have that. They have eyes on the side of their head more, but that's a good position for horses and other animals that tend to be prey animals because they can see around them much more clearly. Whereas a predator, it's better to have the eyes in front with good depth perception. So this is a fixation point on which the eyes are focused. So objects that are farther away, well, they're more medial here on the fovea, and items that are closer, like this one, they're farther away on the fovea. So that's how you can determine how far away something is. And the pathway is quite interesting, the visual projection pathway. So this is the optic chiasm here. And you can see that some of the cells, the ganglion cells, are on the same side, and they go to, for example, the visual cortex in one hemisphere, whereas the other ones from the other eye, the left eye, cross over, and they go to the other one. So what I'm trying to say here is that some cross over and some stay on the same side. So some of these neurons will cross over and some will stay on this side. Yeah, so that, I think we talked about that before. That's called hemidecussation because it's not all of them. So some are ipsilateral and some are not. So the bipolar ganglion cells are first and second order neurons. Hemidecussation happens in the optic chiasm, which you might have noticed with the brain dissection. Half of the fibers decussate so that images of all objects in the left visual field fall in the right half of each retina, and each side of the brain sees what is on the side where it has motor control over the limbs. Third order neurons are in the genticulate nucleus of the thalamus, and they form an optic radiation to places like the visual cortex, where visual sensation occurs. But a few fibers go to the colliculi and the midbrain for visual reflexes, such as the pupillary reflex and accommodation for near vision. So some processing occurs already in the retina. Contrast, even adjustments for brightness, motion, stereopsis. The visual association areas, though, in the parietal and temporal lobes process that visual data. They will determine such things as object location, motion, color, shape, boundaries, and they store visual memories there. So you can recognize the printed word, for example. Yeah, so thus endeth our lecture on vision. Thank you for watching.